Hey guys, and welcome to chapter 12. In this chapter, we're going to talk about viruses, like what it is to be a virus, how are they genomically different, and then four main viruses that we're going to see routinely throughout term, and then also how do they replicate. So let's go ahead and get started. So viruses are not considered living because they have to replicate inside of a host cell. So they can live temporarily outside of a host, but if they don't infect a host cell, either be bacteria or human, they will not continue to survive. So, with that, an example is the measles virus. So, the measles is probably one of the ones that we've been hearing a lot about, especially in the news with the whole anti-vax movement and everything. So, this is actually a image of a virus being shed from a host cell. So within a virus, some of them they have a capsid and then a viral envelope, and we'll go into what that is very briefly. So not all viruses are envelope, but they all have capsids. So different viruses have different forms of mechanisms in which they like to be transmitted. So like HIV, human papilloma, HSV, those all prefer to be direct contact and primarily through sexual contact. But if it's just direct contact where it's like skin to skin, it is possible for that to happen, as well as with skin to skin contact, it includes like bodily fluids, whether it be blood or vaginal, semen, spit. That all includes direct contact. Whereas like measles, like they prefer to be like in droplet form, so that's a lot more effective because you don't have to come in contact with somebody who is infected at the time. And then also host range, that will be determined by what species um, will be infected. So, like the flu specifically is a lot more broad in their host range because it can infect swine, bird, human. And then with like HIV, it's a little bit more narrow. It's still considered broad because it can go between humans and then like monkeys, but it's still a lot more narrow compared to the flu. Human papilloma is actually only infecting humans. That's why it's called human papilloma. Okay. So viruses, they are pretty small compared to other genomes, but also there are some consequences of having a viral infection, primarily with like medical therapies. So antiviral agents are very, very hard to discover. There's very few targets, meaning that it's not as if you can target a cellular membrane like how we do with antibiotics. So it's a little bit more difficult to develop a drug. And then also antiviral agents, a lot of times they have very, very severe side effects. But as we discover and as the drug community develops, we are beginning to produce a little bit more better antiviral medications, but it's still very harmful to our patients, and we don't like that. And then also, viruses, their genomes, they mutate and replicate extremely fast, even faster than bacteria. So, meaning that with them constantly mutating, an antiviral medication may not work every year. That is partly why we always have to get a flu vaccine every year, because the flu virus is always mutating over time. Okay, so now with that, we're going to go into some viral structures. So viral genome is either DNA or RNA based and single or double stranded. And then also all viruses, they'll have a capsid. And this capsid is like their version of like a nucleus that actually encloses their genomic information. So it's a protectant. And then also sometimes some viruses will have an additional layer called an envelope, but the envelope is directly derived from the host. So some viruses, they won't develop an envelope because they haven't come in contact yet with with the host cell in order for them to integrate some of the membrane into their structure. So there are four main biostructures that I want you to be able to recognize, and the first one is isohedral capsids. So they are perfectly symmetrical. They have 20 faces, triangular faces, and an example of those is herpes simplex. There are some other ones such as like polio. Those also are isohedrals as well. But I'm more concerned about that that is perfectly symmetrical. It has 20 identical faces. An example of it is herpes simplex. So the next one is filamentous. So this is where they are helical in shape, but they are perfectly symmetrical as well. It kind of creates a tube around the genome. And in the picture, the red part of the top image is the genome, and then the blue part is kind of like the capsid part that is protecting it. And then can be very, very long, and it is somewhat flexible. It's not very rigid, but it's still rigid enough where it can protect the genome. And an example of that is Ebola. Okay, our next one, our third one, is a bacteriophage that is tailed. So this one, they also have a isohedral head, but they also, on top of just having the isohedral head, they have a tail structure. So this tail structure, we've sort of seen it with transduction, where a bacteriophage will attach to a bacteria or a host cell. So this part is for attachment, and specifically a delivery system. So it can deliver the genomic information into a host cell. And then the last one is amorphous. So amorphous, they have no true structure. They're not symmetrical like the other ones, but they do have a envelope with it that does protect the core of the genome. And then an example of that is smallpox. Okay, so now we've talked about the shape. Now let's go into how do they arrange their genetic information. So as I said before, viral genomes, they are very, very small, but they evolve very rapidly. And with that enables them to invade a host defense very, very quickly. And then with all the mutations, sometimes they can even jump into new host, like jump into an entirely new species that they weren't able to infect before. But a lot of times they are very, very simple. So the one above is a RNA-based virus. So it has three small genes that we can see: the GAG, the POL, and the envelope. So the GAG is specifically for the core of their genome. The POL is for the enzymatics, for the enzymes, and then the ENV is specifically for the envelope that they're going to produce in order to protect their genomic information. So I'm not going to expect you to know specifically what those genes do or what do they regulate. I just want you to know that the viral genome is very, very simple. And then the fact that they're so simple and so small, they can evolve very rapidly. And with them evolving very rapidly and mutating, it can enable them to jump from species to species, making them more virulent and more of a threat in a way. Okay, so with that, we're going to take a little sidestep into prions. So I don't know much about prions, but I do know the basics of them. So with prion diseases, what happens is that once you get a prion disease or a prion virus, what happens is that the prions, they'll produce an enzyme that will cause misfolding of proteins that are already existing in cells. And then with the misfolding of proteins, it will eventually, all those misfolded proteins, they will accumulate. And then with that can cause a lot of harm to the host. And especially if, like in humans, it can cause harm if it's a prolonged prion disease. A lot of times it'll lead to a lot of cell death. And then with a lot of cell death in the neuronal areas, so like in the brain, it can cause like disorientation and even dementia, and then eventually they have minimal to no cognitive function. Okay, so that's pretty much what I actually just said. So this is like the normal way our protein would look. But then if we have a prion, what it does, like I said, it'll produce that protein enzyme, and it will cause a abnormality in its shape and structure, and with that, that can lead to um, harm to the host. Okay, so with that specifically, how do viruses get their diversity and how have they evolved? So they have evolved because their genome is very, very small. That is why they can become very diverse in a very, very short um, frame, time frame. And then also with that, the fact that they replicate so quickly, they get a lot of random mutations. And with a lot of those random mutations, that can lead to something that's called angenic drift. So this is when a population of viruses, they have this mutation, and then with having that mutation, host cells can no longer recognize it as like a threat. So that's why it's able to infect so quickly, because we don't have any antibodies against it. And we'll talk about antibodies more in 2 chapters after this. But angenic drift will specifically generate pretty much a whole new virus. So that's primarily why like influenza is always having to get new vaccines, because it constantly undergoes angenic drift. Okay, now a little sidestep into the herpes simplex virus. So this is a phylogeny tree of the human and animal herpes virus based on their genetic sequence. So based on their genetic sequence, that we can group them in accordance to their genome and do some comparative analysis between them. With all the herpes viruses, we know that they have a common ancestor. And then as it has evolved and diversed, it has diverged into different categories. And with them diverging, they can now infect across species, primarily though, what's it called? In mammalian species. So, but the most common ones that we'll see is the varicella zoster, herpes simplex 1, and herpes simplex 2. We'll talk more about actually those 3 strains specifically whenever we get to the chapter of infections of the reproductive system. Okay, so how do we actually categorize viral genomes? So there are 7 groups. So they are divided up amongst whether if they're DNA or RNA based, and then as well if they are single or double stranded. So I'm not going to expect you to actually know all 7. I want you to know specifically, if I tell you like HIV, what kind of genome does it has? Is it RNA or DNA based? And I would want you to tell me from that. So with that, let's go ahead and go into some little vocab, and then we'll go into actually specific viruses, and then also what is their genomic base. So before we do that, let's get some basic foundation out of the way. So a general viral replication cycle has to go through these 4 stages. So the first one is that the host has to recognize that it's a virus, and the virus has to be able to attach and adhere to the host cell. And then once it adheres to the host cell, what can happen is genomic penetration. So this is when the virus will integrate its genome into the host cell and gain access to the cell machinery. So it can actually start making more of itself. And then once it gains access, it'll start assembling progeny. And then that's whenever you're making more viruses. And then eventually, once all the progeny are made, they have to be able to exit and then continue transmission. So depending on how they'll exit will determine between which virus. Sometimes they lyse the entire host cell, or sometimes they'll just exit through it through the cellular membrane. But that will be dependent upon what virus. And then once they exit, then they have to be able to infect new host cells. And then the cycle just goes on until the infection is done. So this is specifically the attachment and the genomic penetration part. So this is a bacteriophage that attached to a bacteria, and then it has adhered to it. And then once it's adhered, it can drop off the genomic payload into the host cell. And then once the genomic payload is in there, then the virus genome can start replicating. Okay, so with that, that brings us to two viral cycles. There's the lysogenic and the lytic. So the first one that we're going to talk about is the not the lysogenic, sorry guys, the lyse. So this is where it will enter the cell, it will replicate, and then it will burst the host cell and then just spread all of its progeny. Whereas the lysogenic, what will happen is that it will enter the host just like normal, but instead of making more of itself and lysing the cell, what will happen is that the viral genome will integrate itself into the host genome, and then with that, the host cell will just continue on replicating how it normally does, but it will continue to have the viral genome inside of it. Eventually, sometimes what will happen is that a cell will get a signal to go into the lyse cycle, which is perfectly fine, and then it'll follow through the lyse cycle. And I'll show you a figure of that in one moment. But one last thing I want to point out is what a prophage is. A prophage is whenever the viral genome is integrated into the bacterial genome. So that's whenever you have two different genomes in one. So that's called a prophage. So here is the graph or figure that represents what we just talked about. So the first one right here, this is the lytic cycle. So this is where you have a host cell, a phage attaches, and then it will insert its payload, and then from there it will replicate more viral progeny, and then eventually they will lyse the cell, and then you'll just have a lot of phages. Now, if it doesn't go through the lytic cycle, it'll go through something that's called the lysogenic cycle. So this is where the viral genome and the host genome will integrate into one, forming a prophage. And from there, what will happen is that the cell will just continue to replicate, so on and so forth. And then that is how the virus will live on in that era, which is where it's not considered virulent, meaning it's not considered really causing harm. It is, but it's not causing like an active infection. Okay, so with that, we're going to talk about culturing viruses. So culturing viruses is very, very hard, solely because viruses, they have to replicate with inside of host cell. So if you just have a virus but no host cell for it to infect, you will not get a good viral culture. You may not even get a viral culture at all. So what you need to do is that you need to also have a tissue culture in the presence of your virus. So with that, you either have like animal cells or human cells or even a live animal model that has the virus in it, and then the virus is able to replicate, and then you're able to study it from there. So the most common way is E. coli. E. coli is super easy to manipulate. So what you'll do is that you will have phage, and then you'll have, so like your virus, and then you'll have E. coli, you'll put it into one, and then you'll incubate it so they grow together. And then what you'll do in order to keep the E. coli alive for the virus, you'll pour the E. coli broth onto an agar, kind of like what we do in class, in lab. And then what will happen is that you will have the E. coli being fed by the agar, but then the E. coli is feeding the virus. So you got to make sure that you're providing nutrients for both organisms. Okay, so this was pretty much what I just stated. This is just another picture for it. Okay, so now we're going to go into actual viruses. So the first one that we're going to talk about is the papillomavirus, specifically the human papillomavirus. So this is the general idea of how it will infect. So what will happen is that you'll be exposed to somebody who has HPV, and it will infect the basal cells on normally the genitalia area. And then what will happen is that the virus will replicate, and then once it's done replicating, it will shut off and release HPV virions, and then with that, those can continue infecting more and more cells. And human papilloma virus is specifically a double-stranded DNA virus. Now with human papilloma, it is very, very prevalent in our society. It is super contagious, very, very infectious. It's primarily spread through sexual contact, whether it be oral or anal or vaginal. Just sexual contact in general can lead to the spread of HPV. The most common way, though, to protect yourself is condoms, as well as there is a vaccine for HPV, specifically the HPV that causes cervical cancer. So we'll go more into detail about that, but there are 2 strands of HPV that have been linked to cervical cancer, and I believe it's HPV 16 and 18, but like I said, we'll talk about more of that later on in term. So whenever it comes to these viruses, guys, I want you to be able to tell me what is their general way of gaining entry, what kind of virus are these? Are they DNA or RNA based? And then also, what are some preventions, treatments, anything of that nature? So the big picture of each virus. And then generally, if you are going to vaccinate against HPV, you normally do that before somebody reaches sexual maturity, so they do have the full protection against HPV. And then, like I said, one of the main ways to prevent it is through condoms, abstinence, if that is your choice, and then also monogamy, if that is your choice. And then if you do come in contact with somebody who does have HPV, then just have an open conversation with them. If it's not guaranteed that they'll transmit it to you, but you just need to make sure that you are protecting yourself. Okay, so this is the actual process of which how HPV will replicate inside of a host cell. I'm not going to expect you to know that though, guys, but I do want you to know that it has to attach, it has to be able to replicate, it has to be able to release this progeny, which is with every virus. Okay, so with that, we're going to go into the flu. So this is a flu virus. It's very, very simple. It is RNA based, and it only has 8 chromosomes. So it's very, very simple. And the fact that it's so simple is why it can, one, replicate so quickly, and then as well as each segment has a designated function. So rather than having to divide and conquer between segments, one just each has its own job, which is really nice for influenza, but kind of bad for us. But there are 2 specific enzymes that I want you to know about whenever it comes to the flu. So I want you to know about this NA and this HA. So specifically with like Tamiflu, so Tamiflu is probably like the most popular drug for the flu infection. But what Tamiflu does is that he actually blocks the activity of NA. And the specific purpose of NA is to actually allow the virus to leave the host cell. So the fact that Tamiflu blocks the virus from exiting the cell, the virus can't continue on in its infection cycle. So that's really good. So NA has to do with the virus leaving the cell. HA specifically has to do with it binding to a host cell. So that is how the virus will actually enter the cell. Okay? So with that brings us to HIV. So HIV, he is a RNA based, he's double stranded. He is very, very different than the other viruses because he uses something that's called reverse transcriptase. This is an enzyme that will produce double stranded DNA from RNA. So rather than starting off DNA to RNA, he will start RNA to DNA. So that is why it's called reverse transcriptase. So he's very, very unique in that sense. And then also, something called a provirus is produced a lot of times with him, where you have viral genome is integrated into host genome. And then also with HIV, he has two main ways of eluding, so avoiding our immune system. The first way is what happens is that he will produce a large nucleated cell, and then with the fact that he produces such a large cell, he is able to fuse other cells very, very rapidly, very, very quickly. And then another one is through nanotubes. So this is the small connections between cells, and with that, it aids in communication a lot more. So with HIV, this is what he looks like. He's very, very complex in sense of a virus. But the main thing that I want you to know is reverse transcriptase. That's the main thing that I want you to know about HIV, is that what enzyme does he use, and why is that advantageous? Okay. So this is the actual process of how HIV will infect. Like I said, I'm not expecting you to know his actual replication process, but I do want you to know what does he actually infect. So he specifically will infect T cells, which are directly linked to the immune system. And we'll talk about T cells a lot more when we get to those chapters. So that is how HIV will affect. He'll infect T lymphocytes or T cells. Specifically with this image, you see all these little red boxes. So this is different medications. Same thing with the other images. So there is a red box right here. So these red boxes will represent the type of medicines that are made against this virus and what do they actually do. So with HIV, there are numerous different kinds. Shouldn't use an updated one because we actually have now an HIV medication that is now on the market that is kind of like a plan B, where if you feel that you've been exposed to HIV, what will happen is that you'll take this and that will theoretically eliminate the chances of you getting HIV positive. But so I'm not going to really expect you to know all of these medicines, but I do want you to know that they are out there. Now, with that, we skipped this in lecture, so we don't need to know about this. Okay, so that will conclude our lecture. So in this, we learned about what is a virus, what are the 4 viral structures, as well as their viral genome, how does it make them diverse, how are they diverse, and then also the examples of the viruses, specifically with herpes simplex, the flu, HPV, and HIV. Now, we did talk about very, very briefly their replication cycle, but I'm not going to expect you to know those steps. But I do want you to know the big picture of each flu. And then we also talked about the general steps of a viral life cycle, where they have to attach, insert their payload, they have to be able to replicate, and then also exit and continue transmission. And then the difference between the lytic and the lysogenic cycle of a virus. So I hope y'all enjoyed this lecture. If y'all have any questions, don't hesitate to contact me, and y'all have a good day.